Hey everybody, and welcome to ResearchCon 2024. My name is Cheyenne Hoyle, and I'm so honored and excited to be one of your co-chairs this year. I was diagnosed with cystic fibrosis when I was 2 years old, and now 22 years later, I am a University of Georgia graduate and am currently working at Wellstar MCG Health in Augusta, Georgia, as a molecular technician in the microbiology lab. I can't wait for y'all to see what we have planned.
Thank you, Cheyenne. I'm also excited about ResearchCon and being a co-chair for this year's event. My name is Art Brace, and I'm also living with cystic fibrosis. One of the reasons I enjoy ResearchCon so much is that in the same event, I can learn about the latest in CF research and drug development, new advances in patient care, and can also interact directly with other patients and feel a greater connection to the CF community. As co-chairs, we help put together a diverse set of presentations and roundtable discussions, so hopefully everyone attending will find topics that interest them. There are also opportunities to connect with others in the community through the chat, virtual water coolers, and direct networking. Next, we will hear from the third co-chair, Cheyenne Pye. Yes, it's true. We have both Cheyenne and Cheyenne as co-chairs. I'll try my best to keep your name straight over the rest of our ResearchCon event.
Oh, thank you so much, Art and Cheyenne, for the warm introductions. And what a pleasure to be co-chairing with you both this year at ResearchCon. Hello, everyone. My name is Cheyenne Pye. I'm the pediatric respiratory therapist and program coordinator at Dell Children's Medical Center in Austin, Texas. And on behalf of the ResearchCon Planning Committee, speakers, facilitators, and session advisors, we want to welcome you and thank you for being here for our 6th annual conference. ResearchCon is just so important to us. Whether you are on the technical, clinical, or personal side of the CF community, it really allows us to connect and learn with each other and share experiences in a virtual and safe environment. Conferences like ResearchCon really bring us together in one place. It gives us hope of what to come as we walk down the path to finding a cure together. You really don't have to meet in person to learn new information and engage with those who share the same passions.
Absolutely, Cheyenne. ResearchCon is an open and welcoming environment for anyone to learn more about cystic fibrosis, its many facets, and what we as a community are doing to address these facets moving forward. Our goal is to help alleviate the fear of the unknown and get you, the community, more involved in future research. This year's theme for the ResearchCon is where we are and where we're going. Now, more than ever, the CF community has seen unprecedented growth and improvement to our quality of life. We're not just surviving anymore, we are thriving. We have been able to shift our focus onto reproductive research, the effects of aging in the CF population, and transplants of organs other than the lungs. As a young adult with CF and a member of the next generation of patients, this is incredibly encouraging. Also, as a patient who cannot tolerate current modulators, I'm hopeful of the research that is also being devoted to studying CF liver disease, rare mutations, and other genetic therapies that will benefit even more patients. But as exciting as the future is, it's also important to talk about the relevance and progress of CF research. The community needs to know what they can currently expect from the drug pipeline, as well as what they can do right now to make an impact, such as joining a clinical trial or the patient registry. As we get ready to dive into the science and evolving landscape of CF care, I just want to take a moment to assure everyone, especially if this is your first time joining us for ResearchCon or any CFF event, that it's okay to be overwhelmed. It's a lot of information. I encourage you to make notes, ask questions, and take advantage of new connections during our virtual water coolers. Now, without further ado, please welcome back Art as we get ready to kick off our keynote presentation.
Hello, everyone, and welcome to ResearchCon's opening keynote, Where We Are, CF Drug Pipeline Developments. My name is Art Brace, and I am one of the co-chairs this year, and am living with cystic fibrosis. For our plenary talk today, we will be joined by Dr. J.P. Clancy and Nicole Kohr, who will talk about her CF journey as well. Before we get started, though, and before we dive into the keynote, can we go to the next slide, please? Thank you. Before we get going, I want to provide a few housekeeping items. All the keynotes and information sessions during the next 2 days will be recorded and available after the event on the CF Foundation's YouTube channel. The roundtables and social lounges are not being recorded. If you have questions, feel free to put them in the chat under the Stage tab on the right-hand side of your screen. We may not be able to answer all of your questions, but we'll try to get to as many as possible. We won't be able to answer questions about individual diagnoses or treatment plans, and we encourage you to use the information you learned today to have conversations with your care team. If you experience any technology problems during the event, please visit the help desk in the expo room on the left-hand side of your screen. We also want to remind everyone of our event conduct guide. Please be kind and open-minded and respectful of everyone's opinions and life experiences, and please do not ask for or give medical advice. If you need any staff support at any time or want to report a violation, we encourage you to visit the help desk. Now let's get started. I'd like to tell you a bit about my story and my interest in ResearchCon. I'm 58 and live in Northern California. I was diagnosed with CF at age 12, and I grew up in the Cleveland, Ohio area, and now have lived in California for the last 35 years. I'm excited about the start of ResearchCon. As a patient, I'm interested in learning about advances in CF research, drug development, and clinical care. I'm also trained as a scientist with a PhD in genetics and have spent my career working in new drug development at several biotech companies, so I'm very passionate about research. I've benefited from the new drugs developed over the years, especially Trikafta, and am excited about new drugs in development now that have the potential to reach even more patients. I've also participated in several clinical trials and research studies over the years. My work as a scientist has not been on CF, but for cancer and inflammation, although there is a lot of overlap in the discovery and development process with CF therapeutics. If we could go to the next slide, please. I'd like to show this slide because I feel that it helps to show the complexities of new drug development that I experienced during my career. And this relates to what you will hear today about different types of drugs in different stages of development for CF. The funnel represents how a potential drug progresses from an initial idea in the lab to an approved therapeutic. From the top to the bottom, you can see the different stages that have to happen, from cutting-edge basic science to creating a drug in discovery, to testing it rigorously in preclinical development, through multiple clinical trial stages in CF patients, and then potentially to an approved drug at the bottom. The different colored balls represent different types of therapeutics in development, such as new antibiotics, phage therapy, new modulators, mRNA therapeutics, and gene editing therapies. Why I think this slide is important is that it shows that this process can be very long, often 10 to 12 years from the initial development work to an approved drug. Developing a safe and effective drug is incredibly difficult. Along the way, some of these early approaches just won't work. Some failures expected and failures okay. You learn from all the data gathered, and it helps make the next set of drugs even better. As patients, we need to keep in mind that in the current CF pipeline right now, there are multiple drugs moving forward at all of these stages. Some are in the earliest stages and have years of development ahead, but other new drugs that have already completed the earlier stages have the potential to be available much sooner. I'm excited about the progress of the CF drug pipeline, and you'll hear much more about that shortly. I believe that ResearchCon is important to the CF community so we can better understand the complexities within the drug development pipeline and be hopeful that multiple approaches are underway to find new treatments that have the potential to help everyone with CF. How can patients help with the development of new therapeutics? It's important to be part of the patient registry and to talk to your care team about potential research studies and clinical trials you may be qualified for. Later today, there will be a presentation on what it's like to be part of the clinical trial as a patient. There are many different topics in the presentations and roundtable discussions over the next 2 days, and hopefully everyone can find something that they can learn from or a group they can connect with as part of the CF community. To get us started, we will now begin today's talk. It's my privilege to introduce Dr. J.P. Clancy, the Senior Vice President of Clinical Research at the Cystic Fibrosis Foundation, who will give today's keynote presentation on the drug development pipeline for CF.
Thank you, Art, and good evening, good afternoon, wherever you may be. It is so good to be here. My name is J.P. Clancy. As Art said, I have been with the CF Foundation about 5 years, and before that, I was a CF physician taking care of people with CF and also a CF researcher. And I am really excited to be able to update you on our pipeline of research that's happening right now. It's safe to say we are in one of the most exciting times in CF research and clinical research in particular. And so I feel really honored to be able to share this with you. And I also just want to highlight how this is one of my favorite events that we do every year. I think I've been here again roughly 5 years, and I think 4 of those I've had the chance to participate in ResearchCon. So so good to have you all here today. Next slide. Just a brief disclosure, if you want more information, you can find it on our CFF website. Next slide. And this session, we're going to be spending some time talking about various kinds of drugs and new therapies that are in development. And a lot of it's going to be around therapies that are treating the cause of CF, and that includes drugs like CFTR modulators, and also genetic-based therapies and other drugs that are trying to activate ion transport that may be beneficial for people who are not able to take CFTR modulators. And we'll also spend a fair amount of time talking about CF infections and some of the new exciting treatments happening in that space. And what you're looking at down below is a snapshot of our pipeline, our therapeutic pipeline of research that's supported by the CF Foundation. And we'll spend a little more time on it a little bit later, but the point is each one of those colors represents a different type of therapy, and the taller they are, the further along they are in clinical development. And why I'm excited is because a lot of these are well on their way to later phase clinical trials. And as Art said, some drugs take as long as 10 to 12 years to be developed and become approved. The good news is we've got a head start on a lot of these different therapies, and we'll talk about those in a moment. Next slide. So before we get into the weeds of the research that's happening and some of the drugs that are under development, I wanted to just take a moment and just kind of orient people who may be newer to CF, make sure that you have a little sort of a common understanding of what we're talking about today. And a reminder, I think this community, if anybody knows that CF is a full body disease. And you can see a number of the different parts and organs and so forth that are affected in CF. The common theme or a common problem is a gene that doesn't work, and that gene's name is CFTR. You can see the long name right there. I won't make you go through it again. But when that gene and its protein don't work, you get the symptoms of CF. It's a genetic disorder. In the US, it's roughly 1 in 3,000 births. We estimate about 40,000 people in the US are living with CF, well over 100,000 worldwide. We think that's a pretty vast underestimate because there's a lot of places where CF is not really sought or looked for. And as noted, a number of organs can be affected. And we've also identified a number of different ways that the CFTR gene can be broken that results in disease. And all of these variants or mutations, although they may differ, the common theme is that the CFTR just doesn't work. Next slide. So if we took a look in the lungs, and this is a cartoon, and just to give you a sort of help build out sort of how that problem in the gene leads to the symptoms of CF. When CFTR doesn't work, salt doesn't move correctly. And what we're looking at on the right side is a cartoon of an airway. And we can see that there is this kind of lining of thin mucus that's kind of green, and that's normally just kind of helps sort of trap things and get them out of your airways quickly before they can stir up trouble. And when CFTR is working, salt moves, water moves, that mucus is thin and watery and is able to be cleared. But when CFTR does not move, which is on the right side, the mucus can get thick, it's dehydrated, and then it can cause problems or develop problems with infection, inflammation, which in the long run over time results in lung damage. Next slide. And another way of looking at that is shown here. Starting at the top, we have a gene that has a mistake in it. That mistake leads to a protein that doesn't work correctly. That results in salt and water that don't move quite right and sets up this cascade in the lungs of buildup of mucus, infection, and inflammation. And over time can result in respiratory failure, people having to resort to lung transplant, et cetera. And we're going to talk a little bit about two types of therapies. The one sort of below, those are symptomatic therapies. They're trying to treat the symptoms of CF, treating the mucus problems, the infection problems, the inflammation problems. We'll talk about that in the second half of the presentation. The other part that I'll start with, though, is CFTR therapies. And those are really treating the upstream problem. And again, when we talk about CFTR modulators, we're talking about drugs that help make the CFTR protein work better. And when we're talking about genetic-based therapies, we're trying to take a step backwards further to try to give back a working normal copy of the CFTR gene. Next slide. So before we even get into that, I guess a basic question is, why are CFTR therapies, why do they make sense? Well, what we've learned from CFTR modulators like Trikafta, Ivacaftor, or Kalydeco, that if you fix CFTR, it can be life-changing. And what I'm showing you here is some data that came from the pivotal studies right before the pandemic of Trikafta, basically. And in the study, these were people who had one copy of F508, most common mutation that causes CF, and a second mutation that had no function in it. And what we found is when people started Trikafta, or that combination of ETI, elexacaftor, tezacaftor, ivacaftor, had this dramatic increase in lung function that was sustained for months, many months. And down below, at the same time we saw lung function go up, we saw sweat chloride drop down. And that's a marker that CFTR is working. And this has really demonstrated unprecedented benefits across a lot of different outcome measures, things like lung function, day-to-day symptoms, growth, infection, and even survival. And so the main reason I bring this up is just to say CFTR makes a lot of sense for people with CF. And if we can find ways to give back or restore that CFTR function that they have, we can really, hopefully, have it translate into a big clinical benefit. Next slide. So I wanted to give you a snapshot of what is happening with CFTR modulators right now. And the left side, it highlights all the available CFTR modulators for those who are candidates for them. And I won't go into the details, but the point is they're all, by and large, in studies getting into younger and younger populations, which is very exciting because we think using these drugs earlier may be able to prevent a lot of the problems that can happen over time with CF. I wanted, though, to take a little, just take a moment and highlight you on some very recent data from a new triple combination therapy. And the drug doesn't have a name yet. This is the full generic name of vanzacaftor, tezacaftor, deutivacaftor. Try to say that 3 times fast. And there were basically 2 study groups, the Skyline study, which included people over the age of 12, and then something called the Ridgeline study, which was for children ages 6 to 11. And the study was designed to really compare exactly how this new triple combination compared with Trikafta, and I'm calling it ETI here for just an abbreviation. And what they found was that lung function between the new triple combination and Trikafta, they described it was non-inferior. It would basically meant they were essentially the same. And what we also found was symptoms were very similar, cough was very similar, and risks of exacerbations and so forth. One of the big benefits potentially of this drug, though, is that it's a once-a-day preparation. So it doesn't require that second dose in the evening or morning, however you take your medication, to be able to get the full benefit. So that might be a real improvement for some people. What also is exciting, though, is what I'm showing in that table down below, is looking at the percent of people who had sweat chloride values under certain numbers. And that's what that percent SC means, percent sweat chloride. And the top, we're looking at people who had sweat chlorides in the 2 treatment groups that were below the diagnosis of CF. And what we found was that those who were on the new triple combination, DTD, 86% of people by the end of the study had sweats less than 60. If you go down to less than 30, which is actually the normal range, we found that 31% were on the new triple combination had sweat chlorides less than 30. When we went to younger patients, what was really interesting was that the percent of people with sweats under 60 and under 30 was significantly more. You could see that over half of the people who were children 6 to 11 on the new triple combination had sweat values in the normal range. And we think that over time that's going to translate to continued benefit by having really normal levels of working CFTR. So more to come. This is not published data yet. We've only seen press releases, but we're all very excited about this new triple combination that maybe hopefully will be approved in the not too distant future. Next slide. In addition to that combination, which is under development by Vertex, well known in this field, who've really developed all the CFTR modulators thus far that are approved, there has been some exciting movement with another company. Their name is Sionna, and they are developing a different sort of class of CFTR modulators. And I'm just highlighting a couple of press release pieces of information that they basically were able to raise an awful lot of money to develop this drug, which is exciting. And I showed that, we certainly provided some funding, but the point is there's a lot of excitement about the potential of this therapy to be able to be beneficial. And what's different about it, what we're looking at on the right side is just a cartoon of that CFTR protein that I was describing before. And this company has developed a modulator that really goes after a unique part of CFTR. Its name is NBD1. And NBD1 is where the actual problem with delta F508 resides. It turns out all the other modulators that are available now bind to different parts of CFTR, but not to NBD1. And so this at least opens up the possibility of things like potentially higher efficacy, potential to help benefit additional variants, perhaps a different side effect profile, and also importantly choice. And we think that's a really important part of modulator therapy for people with CF. So I would just say keep an eye on this. They're in clinical trials now, still a little bit early days, but we are really excited that this program has got a lot of support going forward. Next slide. So what does having access to CFTR modulators, what does that translate to in terms of outcomes? Well, what's kind of amazing, I mean, we're really literally about 5 years into the broad availability of Trikafta for a lot of people with CF. And really over that relatively short period of time, we've already seen a significant disparity or difference in outcomes develop, being based on whether people can or not do or don't take CFTR modulators. We're looking at lung function. There's about a 7% difference between people who are and are not taking modulators. We actually see cutting the risk of death by 50%. And we also said they believe that there may be as much as a 20-year survival advantage just now, and we believe that's going to potentially change over time. So the point is, we know that getting CFTR therapies to people with CF is really, really valuable and important, and has really been one of our primary drivers of research for the last 5 years. Next slide. So if we take a little bit closer look at those who are currently not candidates for CFTR modulators, we believe that the best approach for this group in general is genetic-based therapies. And the idea here is that these genetic-based therapies, it doesn't matter what your gene type is. It really just has the ability to benefit essentially anybody with CF. But first studies are likely to be conducted in people who are not taking CFTR modulators. And so on the left side here, what we're looking at is just sort of a breakdown of people who are taking modulators and those who are not modulator eligible. And when we look, the majority of people with CF are at least candidates based on their genotype for modulators. But there's a group, 6 to 7%, who are not modulator eligible because of their mutations. And they're broken down here in that sort of blow up there. And the point is, there's a lot of different types of mutations within that small group of people. And we look at that group, roughly 1,600 of them are adults. And the reason I bring that up is because when we talk about genetic-based therapies, we're probably talking about studies that begin in adults because of the potential for risk-benefit considerations that probably keep us from jumping too quickly into pediatrics. And then we look at development for those who might actually be candidates for those early phase clinical trials, looking at people who have not eligible for modulators and have lung function of at least 40%, not had any lung transplant. We know this group is kind of a unique group. It has a much higher representation of people who are Black, Indigenous, or people of color, Hispanic in the CF community. We know that about 25% of the Hispanic CF community are modulator ineligible, and roughly 33% of Black people with CF are modulator ineligible. And importantly, too, a minority of this group have really had the opportunity to be in clinical trials lately, and that kind of makes sense, unfortunately, because a lot of those studies have been in the development of modulators. And so this is really where our focus is going to be in hopefully starting to bring some of these new exciting therapies forward to people who are not benefiting from modulators today. Next slide. So if you look across the country, where are people who are not able to take CFTR modulators? And basically, the color of each state, the darker the color, the more people who are not modulator eligible. And as you can see, it kind of pops up in areas where you might expect to have a higher number of people of color. California, Texas, Florida, New York, you can see. And each one of those dots represents a CF research center. And the point is, we need to be able to have a research environment where people who are in those places with more folks who are not on modulators have access to participate in clinical trials. And we need to have mechanisms in place so that people can get from those sites that maybe aren't research sites, but get to those sites in an effective way to participate and be candidates to potentially participate in these clinical trials. Next slide. Oh, well, unfortunately, that one didn't translate well, but what we're looking at here, this was some survey work that we did in the just as we were kind of coming out of the pandemic and realized, that we were going to need to develop these genetic-based therapies for people with CF. First questions that we asked was very basic, is there a basic interest in genetic-based therapies in this population? We asked about 550 people affected by CF, and while you can't see it here, it turned out to be about a 50-50 when we looked at both DNA efforts, mRNA approaches, modulators. There was about a 50-50 interest in this. And so really that informed us that we had some work to do to get people ready to consider being in a clinical trial of a new genetic-based therapy, or even just to have a conversation about it. So with that in mind, you can go to the next slide. We started to develop a number of different tools for people to talk about genetic-based therapies. And we also went to our research coordinators and asked them questions about enrolling people who had not been in clinical trials before, people who had historically been marginalized, perhaps people of color, about their experience of enrolling people, these folks, into clinical trials. And we found that most of our coordinators felt like they needed some additional cultural competency training to be able to have these conversations. And they also highlighted how important it was that people heard about these clinical research opportunities from their healthcare provider. And really that has just become a really important resounding message of ensure that we have the tools available so that people can talk about these opportunities with the people they know and trust. And so what I've highlighted here, without getting deep in the weeds, we have a lot of different training opportunities that we have developed. Several of these are for our care team members, several are for our research members, and several of them are for community members to really become more facile and better understand sort of the nuance of these different types of genetic-based therapies. I also wanted to highlight down below that we recently added a clinical trial navigator. Her name is Katie Howe, and she is more than happy to talk to people about participation in clinical trials and help them kind of potentially get connected with a research site that may be conducting a clinical trial that's of particular interest to them. So you can see, again, we have a website, we have an email address for her, and again, she has become much more active over the last several months as her joining the team has become more recognized. Next slide. So when we look across the therapeutic pipeline for CF, there's a lot of different types here. And as we move from left to right, those kind of dark blue, those represent genetic-based therapies. And there's a lot of different types, and we'll go into a little more detail about those in just a moment. In the purple, those are CFTR modulators. The blue, anti-inflammatories. The orange are anti-infectives, and then nutrition is highlighted in the lighter blue. And we're going to spend most of the rest of our time talking about the genetic-based therapies and the anti-infectives. Next slide. So if we zoom in a little bit more to look at the genetic-based therapies that are currently in clinical trials, you can see that there are 5 going on 6, and this is a very active space. And as I highlighted before, the number of people who are potentially able to participate in these studies is relatively small, only around a little over 1,100 folks based on sort of the probably the most likely enrollment criteria. But you can see that 3 of them are kind of focused on mRNA. And again, what is mRNA? That's kind of what has really happened with COVID vaccines. mRNA is really a way to give back instructions to make a normal CFTR protein. And there's 3 different companies that are in this area. So the one a little bit below, gene addition, those are basically using a weakened viral vector or a viral carrier to bring a gene into cells where it can last for a fairly long time, months at least. Again, more to come as we do this, but really the furthest along of those companies right now is that 4D molecular. And then I mentioned one in the middle, which are called ASOs. And ASOs are sort of they're tiny, short little pieces of nucleic acid that really help increase or decrease the expression of sort of a very specific CFTR. And one that's called 3849 plus 10 KB, many people won't know that, but it's a mutation that we know can cause a really very difficult to manage form of CF. And this approach really helps to restore the normal levels of CFTR caused by that mutation. So point is, there's a lot of exciting stuff in the clinic, and all that stuff down below is working to get into the clinic. And that's really where our future is going to take us as we go forward. Next slide. So I just want to give a quick summary of CF and gene therapy. This is not new in some respects. In fact, the CFTR gene was identified in 1989, and by 1993, a gene therapy study had been conducted in people with CF. There's been about 20 clinical trials in CF in the past. Most of these were very small. They were usually happening sort of out of a research laboratory, and maybe 2 or 3 actually had support of an industry partner. And I think by and large, those were not successful for a number of reasons. By and large, it was they were just not terribly efficient, and there was some of them that actually produced some inflammation. So worried that there might be some safety concerns. However, the world's a lot different now today than it was 25 years ago when these studies were really getting started. And really, there's been amazing advances in genetic-based therapies for other diseases. And probably heard some of these stories about advances for sickle cell, bleeding disorders, eye disorders. These new approaches, which I highlight over here on the right side, mRNA didn't exist back then. We didn't really understand stem cells. The whole concept of gene editing or going in with molecular scissors and fixing a mutation didn't exist. And we really have much better ways of carrying these different approaches and therapies to cells in the lung. And on the other piece to remember is we just understand CF a lot better than we used to. And we have much better model systems for testing some of our ideas before we go into the clinic. And I'll show you an example of that in just a minute. Next slide. So when we look at, when we were thinking about airway cells, again, our airways truly start at our mouth and nose and go all the way about 26 divisions out to our alveoli or air sacs. It turns out all those cells, they're not all the same. And I know that may sound kind of obvious, but we're really just now beginning to better understand exactly what these cells do and their specific functions. And just several of them are highlighted here just as an example. The cells that are circled, we believe, are cells that need to have CFTR. One called an ionocyte. It seems to be really important in regulating how salt and water moves across the airway cells. There's only about 1%, they only make up about 1% of the cells in your airway. They make up 50% of the CFTR. So clearly getting ionocytes is important. Basal cells, those are the progenitor cells that sort of give rise to all the other cells. Secretory cells, which also help to secrete fluids. All of those are important in expressing or having CFTR. Ciliated cells, those are the cells that actually help move the mucus. They don't really need CFTR. They have another job in the airway. It's critically important, but it doesn't need CFTR. So what we're learning is we got to figure out which cells get, or which cells need CFTR and how we get the CFTR into those cells. Next slide. I also want to highlight a different approach that is not about CFTR, but could be beneficial for people who are not able to take modulators, and maybe even those who do. And the idea here is that rather than replacing CFTR or fixing CFTR, the idea here is to substitute for CFTR. And really what this company called Cystic Medicines has done, they basically have taken a well-known drug to treat fungal infections, and they've adapted it to potentially help move salt and water more normally. And basically this molecule, amphotericin, can make little pores in the surface of these airway cells, and it can help move ions like bicarbonate and chloride, which we know are really important for CFTR function. And what they've done then is really develop this into a dry powder, and they've really started determining whether it's safe and tolerable, and now looking into studies in people with CF, which really began last year. So the reason I bring that up is even if it's not CFTR, we may be able to develop exciting new ways to treat the underlying problem that uses a different approach, like substituting for CFTR. Next slide. Had to take a drink before we kept going. Finally, I wanted to highlight that, as I mentioned before, it's kind of tricky for us to have a number of different therapies under development for people with CF who are not either not tolerating or are not eligible for CFTR modulators. We really need to make a little bit of a different ecosystem to be able to develop those the drugs in this relatively small group of people. And the idea here is really sort of threefold. One, we would like to be able to generate data that comes from studies. That's what's on the top left in the blue. And those could be from active studies happening right now. And they would all feed into sort of a controlled data group in the middle. That's what that middle blue thing is. Two, we could have data that would come from other sources, not part of clinical trials. Data like from the registry, data that may have come from past clinical trials, data that we can generate in a new group of people with CF. And I'll talk about that in just a moment. But point is, data from different sources, that also can feed into this controlled data set in the middle. And then down below, having a place where that data could be stored and evaluated by both sponsors and regulatory agencies in a very secure type of way. And really building that ecosystem so that we can really have the maximum number of people who are participating in studies actually receiving study drug, and be able to share the control population across different different programs, so that we can really be as efficient as we can possibly be. And so, next slide. One of our strategies is a study called REACH, and you can see it highlighted here, Research Expansion to Advance the CF Therapeutic Pipeline for People with CF Without Modulators. Point, this study is specifically for people who are not on modulators. And the idea here is to enroll these folks, and they will generate data that can be compared to all these different programs in development. And we hope to start this study at the end of Q2, so this middle of the summer basically, and I just read the protocol, and it is ready to get, just about ready to launch. And on the right side, we're going to be piloting a new idea, a website specific to the REACH study, which will really be a place for people to learn about the study and really learn more about research. So, very exciting time. Key point here, it's for people without modulators, ways to get involved in research and potentially help develop the treatments or support the development of therapies that could obviously in the long run could come back and be transform CF for them. Next slide. The last probably 5 to 10 minutes or so, I will highlight some important research that's been happening in infection. And I wanted to highlight, first of all, that I don't know if people remember, but 5 or 6 years ago, we initiated something called the Infection Research Initiative. And basically, this was a $100 million commitment to really jumpstart research for infection. And this initiative included input from researchers, clinicians, drug developers, and members of the CF community, and really came up with some high priority areas, which are shown on the right side. And now I just kind of where we are with this is really just we're at the moment of sort of reflecting, adapting, and focusing what our efforts are going to look like going forward. Next slide. A few sort of accomplishments. There have been over 200 basic science research grants supported. We've hosted workshops with trying to focus on infections by non-tuberculous mycobacteria or Pseudomonas. You can see on the right side, over $150 million have actually been committed to these research projects. Over 60 clinical research programs have been funded. A number of studies really trying to better understand how to manage pulmonary exacerbations, also the impact of drugs like Trikafta on infection, and really sort of trying to jumpstart detection and diagnosis tools that we can use going forward. And I also want to highlight that the therapeutic development portfolio is broad. Over 30 programs have been invested in. And we've really started to develop some new clinical trial data in exciting new anti-infective research like bacteriophage. And I'll show you that in just a moment. Next slide. So I just wanted to remind people, we're looking first here is just a picture showing the prevalence of different types of infections in CF over the years. And what we can see is that staph detection continues to sort of be very common, found in about 70-ish% of people with CF. Pseudomonas tends to be reducing, and what we've also seen is some reduction in other pathogens like methicillin-resistant staph. But what you can see is in those vertical lines, with the availability of Trikafta, we've seen really a downturn in the prevalence of infections across the board. And we're going to give you just an example of that with Pseudomonas that I think is really telling. Next slide. So this is some data that is from the PROMISE study, and the PROMISE study was funded by the CF Foundation, and it really just looked at a variety of different clinical outcome measures in people who started Trikafta. And what we're looking at here is those who were enrolled in the study who had Pseudomonas, and what we looked at was the density or how much Pseudomonas we could detect in their mucus. And what we're looking at on the right side is each one of those lines represents a different person. And on the y-axis, that's basically how much Pseudomonas. The higher the number, the more Pseudomonas. And what you could see is that there was a big drop in the density or how much Pseudomonas on average people had. That's what that red line is. That was really seen over 6 months. And more recent data has really demonstrated that's been persistent for up to 30 months. But what it's interesting too is if you look at the data, it kind of goes, it's sort of a split. There seems to be people who really, maybe even clear infection, which you can see down below, while others who it doesn't clear. And we're trying to better understand what that really means. What are the factors that sort of help people to clear an infection? Does it stay cleared? And does a drug like Trikafta open the door to helping clear organisms by adding additional treatments? So more work to come, but really demonstrating clearly that there's an antibacteria effect of Trikafta. Next slide. I also wanted to highlight, as I mentioned before, the portfolio is quite broad. You can see here some different programs that are focusing on different pathogens. Pseudomonas is highlighted, NTM, types of approaches that could potentially treat a lot of different pathogens. Additional things like fungus, MRSA, trying to protect against ototoxicity or ear damage, and also some of our collaborators. We're not trying to do this all alone. We're working with a variety of different collaborators, both in and outside of industry. Next slide. I wanted to give just a little bit of an update about bacteriophage research. I think people may or may not have heard of bacteriophage, but basically these are viruses which attack bacteria. They don't hurt us. They don't do anything really to us. And this is a war that's been going on forever between the bacteria and the bacteriophage. But there has been a lot of interest in trying to determine whether we could use phage to treat CF infections. We have funded a number of basic and clinical research programs that are highlighted above. But I also want to highlight in the middle that we have generated data from 2 clinical trials in people with CF. The 2 companies are listed here, trying to treat Pseudomonas infection with different phage cocktails or different groups of phage. And so far, what we've been finding is that generally safe and well tolerated. short dosing at this point, only up to about 10 days. No resistance has been seen, and we have seen a moderate drop in Pseudomonas density. So the exciting thing is it looks like we're seeing some evidence that they are killing bacteria. We're next going to be taking steps to find out how they translate into clinical benefit. So a lot happening, but this is really exciting work that has just been coming out of clinical trials this year. Next slide. I also wanted to highlight another approach that is in the clinic. One approach is to disrupt biofilms. And what is a biofilm? Well, it turns out when bacteria are growing as a team, they can release a lot of sort of gunk that helps protect them from either the host, like my immune system or a person's immune system, and it also protects them against antibiotics. They basically are able to keep the antibiotics away from the organisms. What you're looking at there on the right side is just a biofilm that was actually taken from a catheter, and that's Pseudomonas. Staph aureus that's growing on it. But the point is, there may be a way to break up those biofilms, and a company that's trying to do this, their name is Clarametics, and they're basically trying to determine in an early stage study, can adding this therapy actually help a well-known drug like tobramycin actually work more effectively. And you can see here it's kind of an early phase study, making sure it's safe, it doesn't really cause any sort of unexpected side effects. But the exciting thing is if it works, it could translate into lots of different types of infection. So starting with Pseudomonas, but if there's evidence of activity, we are quickly could translate into other types of infection. Next slide. So I am getting ready to tie up, and I'm going to be passing it on to my partner, Nicole, who's going to have lots of really important and meaningful journey to share with us. But I just want to leave you on a few thoughts. As we head into this year, we are really trying to bring CFTR and other ion transport therapies to everybody with CF. We are really trying to better understand infection for people who are on modulators and advance new treatments for people with infections who do and don't have CFTR modulators. And we also appreciate that the way that we develop new drugs for people with CF is changing. And this is making a new ecosystem that includes our industry, regulator, and community partners. So thank you so much for your attention. I'm happy to answer anything that we can at the end, and I'm going to pass it on to Nicole. So thank you, Nicole. Take it away.
Thank you so much, JP and Art. That was a lot of information, and you condensed it so eloquently. So thank you so much. Hello, everyone. My name is Nicole Kohr, and I am a 31-year-old adult living with cystic fibrosis. I am also a bilateral lung transplant recipient as of 2019. I was born and raised in New Jersey, but I am currently residing in North Carolina, where I am the volunteer engagement chair of the Central Eastern Carolinas chapter. So woo, shout out to my chapter. My goal here is just to wrap up this keynote. And I thought it was very important to talk about my love for ResearchCon, its importance, its impact. But to do that, I have to admit something. I was not always a ResearchCon fan. I know. I thought, based on assumptions and judgment, that you needed a prerequisite, right, to understand and benefit from this type of con. You need a PhD, an MD, and as you can see, I don't have any letters at the end of my name. So why would I enjoy ResearchCon? I also believed, because I'm a minority in this community, that none of this information would relate to me, right? I was never someone that was on Trikafta at the right time. I didn't qualify. I was diagnosed at age 5. I have rare nonsense mutations. Always a minority. But here at ResearchCon, we are all equally beneficiaries of this incredible information. So we have to be like a sponge and soak it up, regardless if you're a patient, if you're a clinician, if you're a researcher, if you're a friend, a family, a mom. You need to use these tips and tricks as tools in your toolkit so that we can eventually make CF stand for cure found, right? It's just a vicious cycle of we need to report things that we're hearing, and we need to take all these tips and tricks to help us in our daily life. With that said, though, next slide, please. I also wanted to tie in our overall concept of where we are by reflecting on where we were. So if you look at the picture on the left, that is a picture of me newly diagnosed with CF. I was 5 years old in 1997 taking Pulmozyme. Pulmozyme and tobramycin were the only medications for cystic fibrosis on the market at the time. If you look at the picture on the right, you will see there is a plethora of treatment options. Now, granted, when I was transplanted, it's because I was highly resistant to all medications. And that cycle will continue due to superbugs, but that's why ResearchCon is so important. So again, dive in. It's a lot of information, but soak up whatever you can. So with that said, I would like to tie up with some more housekeeping items. Please fill out the satisfaction pool, which is under the pool tab on the right-hand side of your screen. And right next to the stage chat, there are many wonderful resources related to this discussion, including slides from today. And I believe some are converted into Spanish, if I heard correctly, which is wonderful. Diversity and inclusion, we love it. We love to see it. Please spend some time exploring all of these documents. I'm so thankful to the CF Foundation for this information, for these resources. Coming up next here at 7:35, so starting now, we have 3 options for you. We have CF newborn screening, a landscape of genetic therapies, and the therapeutic lab roundtable discussion. So thank you so much for joining us, and everyone enjoy ResearchCon. Have a wonderful night.